Now, this is the journey of the plant that I have shown. You see from here we start and we see the kind of compounds which have been produced. And each compound which is listed here has been further potentized, further synthesized, derivatives were made, and a lot of drugs have come out of these molecules. These are different years which have been given. You see, the first analgesic pain-relieving drug came from Papaver somniferum, the latex of poppy, opium. And that's the analgesic pain-relieving drug, no match till today to morphine. Terminal cases of pain, only morphine is given by intravenous route. And there are 70 derivatives which have been made to remove the addictive effect of this morphine and potentiate the pain-relieving properties. Quinine has a very unique history, antimalarial quinine. In Peru, prior to 1800, Viceroy of the Peru, his wife was sick, suffering from fever. And her fever was not giving so many things. After all, she was the wife of the Viceroy. One day, a quack was contacted. He said, if a muddy water from this pond she drinks, she will be all right. And the Viceroy agreed, okay, she will drink this muddy water. She drank the muddy water. Third day, she was perfectly fine. Fever was gone. In 2-3 years' time, they realized what is in the muddy water. Are these some kind of inorganic material or something which cured that fever, which was incurable? Ultimately, they found that there are certain trees which are growing on the sides of that pond. And those trees were later on named by Linnaeus as Cinchona, because the wife of the Viceroy's name was Cinchona. It was named Cinchona. This is how the Cinchona was discovered. And subsequently, when chemistry was worked up in 1803, the quinine was discovered, and that was 1803. It became a drug as an antimalarial drug in 1820. It took about 17 years to become a single molecule. Because up to the age of 18, we had been using just the plants as such. As the chemistry advanced, the people started working into the chemical constituent secondary plant metabolites. You see, the plant has its own life system, life cycle, and the plants has primary metabolites and secondary metabolites. Primary metabolites for the growth of the plant, secondary metabolites for the protection of the plant. And those materials, secondary plant metabolites, are for the health of the plant as well as for the health of the human being. And if you see the metabolic pathways going into the plant system, so exciting that it's comparable to the human system. All those N-oxidation, hydroxylation, then glucuronization, all these things are taking place in the plant system as they are happening in the liver of the human body. It's quite exciting. So therefore, these secondary plant metabolites came out to be very, very potent. They were present in small concentration, but very potent drugs. Primary metabolites, of course, now the future is coming for looking into the primary metabolites from the plants for building immunity and strength. Anyway, that's beside the point, but the journey which has gone from the plants to the single molecule. Now, single molecules from 1951 to 1975 was the golden era of discovery of the single molecule. It was called a silver bullet theory of drugs, that you attack with a bullet, single bullet, and you cure the patient. That was the antibiotics, because the flora of the microbes was screened. As the plants were screened, higher plants were screened, lower plants were also screened, and the antibiotics came. And that was the silver bullet theory, with which you kill the microbe, particular microbe, use the antibiotic and kill them. So that era is also now vanishing in the sense that a lot of resistance is developing, and that's a big issue. The resistance to the antibiotics is a very, very serious issue. We are looking for now products which can be used in place of the antibiotic, something else. Then comes the biologicals, protein materials, very difficult to work with, but those are entering. Vaccines and serums, all these things came. So from 1951 to 75 was a golden era of drug research, when maximum molecules were isolated. And if I refer to one of the molecule which was worked in India, but drug was developed by outside world, that was Wolfia serpentina, Sarpgandha. Sarpgandha was worked up at Tibia College in New Delhi. And this, there was a Ajmal Khan. Ajmal Khan was the principal, and at that time the research was going on in the colleges. And Ajmal Khan was a battery of scientists who was working. Siddiqui was one of the scientists who was working on Wolfia serpentina, Pagle ki dawa. This was and he isolated some compound like ajmalin, ajmalacine he gave the name on his guru's nomenclature. But a team of scientists from Switzerland, they heard that there is some kind of Pagle ki dawa which is available in India, but it's becoming very popular, and some scientists are working on it. So the team from Switzerland, Ciba-Geigy, now it is called Ciba-Geigy, at that time it was Ciba. Ciba-Geigy team came to Dr. Ajmal Khan's lab, contacted Siddiqui, and Siddiqui said, this is the plant I am working on. He explained everything. There was no question of patent at that time. So it was 1950s. So in 52, that team visited, and they said, can we take away the mark that you have ejected? I said, yes, you can take it. They took in 1953, they came out with a drug Serpacil. That was a reserpine. So from Serpacil, that Serpacil was the first anti-hypertensive drug which came into the world market. And they minted so much money. The profit they got out of it, Ciba-Geigy Research Center was established in Bombay with that profit. You can see it's just from one molecule. And that's the history of our reserpine, which we lost the game because at that time there was no provision how to characterize the compound. And later on, Siddiqui said, I met Siddiqui at CMAP. He came to deliver a lecture here. And he said, look here, Dr. Handa, I was working on this. I isolated reserpine, kept in my drawer that I will request somebody to get me the NMR and mass from outside so that we could characterize. But within a year's time, Ciba people, they came out with reserpine. That's how the scientists sometimes, facility is not available and lose the game. Anyway, this is just a short citation which I wanted to give with respect to the fate of the scientist. And the scientist should not discourage in such situations. After all, if quinine came, it was for the benefit of mankind. And scientists should think elevated, that something has come out and has been used by humanity, suffering humanity. But what happened at that time by Britishers? Not even a grain of quinine was available to the Indian patient who was suffering from malaria. You see, the quinine was found, cinchona was found in Peru, in Peruvian bark, that was called Peruvian bark, Cinchona succurubera and Cinchona lageriana. And that cinchona was brought to India in Darjeeling, Mungpu. Plant grew very well. And Mungpu factory was established to extract out quinine. And trucks a load of quinine was produced for military of Britishers who were fighting. And that whole quinine was taken away from India. Not a grain was available at that time to the Indian patient who were suffering from malaria. That is the history of quinine, how a molecule coming from plant. Of course, subsequently quinidine came, which was an antiarrhythmic drug from the same plant. So I have listed each compound has a history background. I won't go into the detailed history of each compound. So you see how our anti-cancer drug was discovered from Vinca rosea, Catharinthus roseus. I hope when you get bored, please let me know, I'll stop. because the things when I narrate, I myself get so excited that I forget the time, I forget where I am. So please remind me then, now stop, I will stop it. Now the Catranthus roseus, you know the Sadabahar plant, very common weed. Eli Lilly company, I was in US at that time. So Voda was a group of scientists who was working with Eli Lilly company. They were working on anti-diabetic activity of the Catranthus roseus. And they isolated certain compounds. They found there is cytotoxicity of some of the compounds. And they said, oh, this is something different coming out. Let us forget about diabetes. Let us focus on the cytotoxic activity of these compounds. This is how vinca-leucoblastin, vincristine was discovered. So the first anti-cancer drug which came into the market somewhere in 1954-55, that was vincristine, vinca-leucoblastin, Hodgkin disease, as well as for leukemia. These two drugs were the ones, and subsequently, of course, the Taxol came from your Texas species, and then the Paclitaxel and other things came. So every compound that I've listed in red has a background history. Historical is interesting for the scientists to read sometime how these compounds were discovered, how Sir Turner discovered the compound. You see, anyway, how aspirin was discovered. You see, the scientist who discovered aspirin, his father was suffering from joint pains, very severe pains. And that scientist was working with the Roche company. And he said, why my dad is suffering? I'm working in a pharma company and my dad is suffering from pain. I will discover something. He came across a plant and he found that there is a large quantity of material available. At that time, salicylates were very, very well known. And he came across salix plant, and he extracted acetyl salicylic acid. Now, this acetyl salicylic acid, he esterified and found the aspirin, bring the aspirin. That aspirin, now you see the use of aspirin discovered at that time and has been so much useful. But of course, we have now side effects coming. COX-1 inhibition is a aspirin. Anyway,